Greetings and welcome to a brief history of the biological control of Canada Thistle using the rust fungus Puccinia punctiformis. I'm Karen Rosen, Biocontrol Specialist at the Colorado Department of Agriculture and Canada Thistle Project Manager at the Palisade Insectary. Canada Thistle, or creeping thistle as it is often called, is one of the most troublesome weeds of agriculture in the U.S. It's found in diverse habitats. It's a perennial with a deep extensive root system, which makes it difficult to control with chemical or mechanical methods, but also makes it a great candidate for a systemic biological control agent. After abandoning other less effective biocontrols, the Colorado Department of Agriculture started using the rust fungus in 2013 with the help of Dr. Dana Berner, a plant pathologist at the USDA ARS Foreign Disease Weed Science Research Unit at Fort Detrick, Maryland, where he has since retired. Him and his assistant, Emily Smallwood, have published numerous papers and conducted extensive field research on Puccinia punctiformis. Dr. Berner has made several visits to Colorado and provided a wealth of information and assistance in helping us get our program started. This is a list of attributes regarding Puccinia punctiformis. It is a pathogen. It is a systemic root parasitic fungus. It is host-specific and has no alternate host. It occurs throughout the range of Canada thistle. It is naturalized in the United States and probably got here about the same time Canada thistle got here from Eurasia. It's an obligate biotrophic rust pathogen, so it is entirely dependent on living Canada thistle plants to complete its life cycle, and it's a cause of systemic disease that can ultimately kill the plant. Puccinia punctiformis has a complex life cycle with 5 different spore types. At the end of the slideshow, I have included a diagram of the life cycle if you would like more details. But in this slide, I'm going to briefly explain how it works. So the rust fungus has 2 different kinds of infections, local and systemic. Systemic infection begins after germination in the fall in the root system, but it can be identified above ground the following spring as a yellow, sweet-smelling spore on tall, spindly stems, as you can see in the first 2 pictures on the left. Late spring, early summer, the spermagonia give rise to the dark, rusty-colored spore, the aciospore, on the same stem. The systemic infection ultimately kills the infected stem, usually before flowering, so the plant dies, but the spores are transported by multiple ways, including wind, water, insects, mammals, and mowing to healthy neighboring plants. In the fall, the neighboring plants develop teleospores and can be identified by local lesions on the bottom of the leaf surface. These are the spores that are responsible for systemic infection after germination on newly emerging fall rosettes. Teleospores are also collected in the fall and used for starting new infection the following fall at new Canada thistle sites. Colorado was the first state to use Puccinia punctiformis as a biocontrol on a large scale. We have over 200 monitoring sites that have been established across the state. Large infestations of infected Canada thistle patches, called nursery sites, have been discovered or created over the years. These sites are used for collecting spores and redistributing them to end users. Hundreds of participants have received packages of spores beginning in 2014. Those recipients include farmers, ranchers, producers, land and homeowners, as well as state and federal government weed managers. Just to name a few, we have worked with BLM, Forest Service, state and federal parks and wildlife, conservation districts, county and city weed managers, and research universities. The Colorado Department of Agriculture Canada Thistle Project has primarily been funded from federal grants. The Palisade Insectary has been ideal for implementing the 3 grant-funded projects that I'm going to review next. The first is the biological control of invasive pests, or BCHIP, in collaboration with the Forest Service. This project allows us to use our many resources to reach our out-of-state partners and share our many years of experience and help them accomplish their biocontrol goals. The Organic Research and Extension Initiative, or ORI, has allowed us to interact directly with local organic community that is eager to control Canada Thistle because they are typically limited to few weed controls that align with organic certification regulations. And finally, Applied Research and Development, or RDEP, in partnership with Utah State University. The Palisade Insectary has worked with many universities around the country, collaborating on numerous biocontrol projects. So let's get started. Carol Randall from the U.S. Forest Service has been instrumental in implementing the BCHIP objectives. There were 4 main project goals. The first goal was to implement Canada Thistle biocontrol, Puccinia punctiformis, to neighboring states. We began implementation in 2017, and we currently have 12 participating states. The second thing was to provide training and share information. We were able to accomplish this with several state visits, including visits to Utah, Nevada, Montana, Idaho, and Wyoming. We provided information on the life cycle, instruction on how to identify the rust fungus, inoculation and collection procedure. We have and continue to have quarterly conference calls and unlimited phone and email support. The third goal was to provide protocols for monitoring and spore inoculation and collection. The CDA has various monitoring protocols for different land use, and SIMP monitoring protocol was used by many states. We also developed spore inoculation guides and spore collection guides and made these available to participating states. And the final goal was to provide spores to participating states and help develop nursery sites in each state. I'll talk a little bit more about this in the coming slides, but we were able to provide over 20,000 grams of ground up leaf matter bearing spores between 2017 and 2022. This map highlights the 12 participating states in purple and the first year they received spores from Colorado. Most of the states have reported successful establishment using the telio spores collected in Colorado. As you can see from this bar graph, our out-of-state cooperators receive spores collected in Colorado every year. Spores were sent out each year, typically in September, and accompanied by a USDA permit. This table shows the year each state requested spores and the total spores received after 6 years. As nursery sites are developed in each state, these numbers should decrease. And the last thing I want to mention about the BCIP grant, again thanks to Carol Randall for her extensive outreach, we were able to interface with multiple agencies, including several state departments of agriculture, U.S. Forest Service, BLM, USDA APHIS, and multiple out-of-state universities. The next grant-funded Canada Thistle project is the Organic Agriculture Research and Extension Initiative. It was established with interest in research, education, and outreach. So first thing, we needed to find and connect with organic farmers, ranchers, and producers in Colorado. We acquired many cooperators through the Colorado Organic Integrity Database, where hundreds of names of certified organic operations are listed. In 2020 and 2021, we sent out a public service announcement introducing the biocontrol via email that reached approximately 300 operations on the list. We gave numerous talks and workshops, some of which were specifically targeted to organic producers. We used our Request a Bug website. We made Facebook posts on the Colorado Department of Agriculture page. We contacted all 64 county weed managers starting in 2015 and introduced them to the biocontrol agent Puccinia punctiformis and since 2019, many weed managers were informed of the ORI grant and asked to share the information with organic growers in their counties. We got cooperators by word of mouth from other organic growers. For example, we have 5 or 6 organic growers in Hotchkiss, Colorado, within a few miles of each other that all use the rust fungus. The other program managers at the Palisade Insectary often talk to their cooperators about biocontrols available, and some of them were organic producers. And finally, our out-of-state partners that have received the spores over the years have the opportunity to share them with organic growers that have had few options for controlling this problematic weed in their state. Dean and Susan Vidal have been instrumental in connecting us with organic growers and ranchers in the Durango area, as well as connecting us with students and faculty at Fort Lewis College. They operate Brightwood Farm, a small organic farm outside of town, and belong to the Animas Valley Grange, where dozens of members are organic growers, promoting and supporting the local production of food. Brightwood Farm has participated in various inoculation experiments involving Puccinia punctiformis. This investigation involved inoculating 6 separate transects over a 6-week window in the fall. They recorded temperature and dew each night during the investigation. In 2022, Dean used his tractor and spray tank to test spreading spores suspended in water over a pasture infested with Canada Thistle. Dean and Susan are photographed here working with 2 Fort Lewis College students hired by the Colorado Department of Agriculture to work directly with organic farmers in the area. They were able to map infestations at several farms and conduct monitoring projects that may be followed up by students in the future. 2 field workshops were presented in 2021 to an environmental studies class from Fort Lewis College in Durango. The workshops were located on the organic cattle ranch of one of our end users. The pasture on the ranch had been inoculated with teleospores a year earlier, and systemic infection in the Canada Thistle patch was widespread the following year. The students learned how to identify the rust and participate in spore collection. The same pasture at the James Ranch is also the location of 6 large permanent monitoring transects created in 2021. The transects were designed to measure the change in stem density over time after the widespread establishment of the rust fungus. The pasture is mowed 1 or 2 times a year and grazed by cattle that are rotated out every few weeks throughout the year. We are also documenting the spread of the rust to other pastures as the cows move from field to field. 2 years of data have been collected with plans to continue monitoring for 5 years. Another class was introduced to biocontrol and the rust fungus at an organic tomato farm in Durango. The adobe house also had a Canada Thistle patch that had been inoculated, but no infected stems were observed the following year. We set up an experiment involving a spring inoculation using teleospores on the edges of the greenhouse where the Canada Thistle was the thickest. We'll check back in the spring of 2023 to see if we have achieved establishment. This is one of 3 workshops presented at the Rogers Mesa Organic Agriculture Research Station in the past few years. This one focused on bindweed mites, Canada thistle rust, and Russian knapweed insects. All 3 agents are established at the research center, creating a perfect opportunity to show the participants how to identify the biocontrols in the field. Sonia Daly and I conducted a similar workshop in 2019, and Dr. Dana Berner gave a presentation at the research center in one of his visits to Colorado in 2019 entitled Sustainable Control of Canada Thistle. The Palisade Insectary Canada Thistle Project has a history of long-term monitoring using Puccinia punctiformis that extends beyond the typical 3-year grant cycle. This is just one example of 2 monitoring transects set up in 2014 and followed through 2022. This map shows our ability to reach organic producers across the state of Colorado. I'm going to end with a very quick description of the Utah State University ARDP grant. It was a 3-year multi-state project. The goal was to identify environmental conditions associated with systemic infection of Canada Thistle in the Intermountain West, and also to assess the potential of Puccinia punctiformis in combination with other control tactics as an integrated weed management approach. We were able to share monitoring protocols and data, inoculation and collection procedures, greenhouse experience, and in-person field training in Colorado and Utah. We also provided USU with teleospores for monitoring transects outside of Colorado and experimental greenhouse inoculations. Using the protocol that was developed between USU and the Colorado Department of Agriculture, we have collected 3 years of data from 4 monitoring transects across the state. These established transects may also be monitored beyond the 3-year cycle of the grant, as they are another example of our ability to conduct long-term monitoring projects. We also have a 5th site in the northeast corner of Colorado that is investigating a combination of biocontrol with other control tactics for an integrated weed management approach. Thank you for your interest in this important and crucial biological control agent for Canada Thistle. And I'd like to acknowledge and give special thanks to Dr. Dana Berner, Joel Price, Carol Randall, the Palisade Insectary staff and Broomfield staff, and all the weed managers and landowners of Colorado and beyond. And most importantly, a special thanks to Dr. Dan Bean for making it all possible.